welcome to the second session of Science Symposium 2019. And for our second speaker, we have Ralph Zehr. Ralph Zehr, MD, is a retired radiologist with a continued interest in molecular cell biology and how the new developments in this field contribute and correlate with our understanding of the origin and evolution of life. He spent his entire career in a teaching research hospital. Lecturing on biology and other scientific topics are a continuing interest for Ralph. He held an academic appointment for many years and published many scientific papers. He and his wife Betty are both long-term Urantia book readers, and continue to enjoy reading together and sharing new insights, particularly on scientific topics. Ralph says it is a privilege to be participating in Science Symposium 2019. Thank you, Ralph.
Thank you very much, Nigel, for that very, very generous introduction. You've just listened to Marjorie Ray's excellent presentation, and when we were setting up this schedule, I knew I was taking a serious risk to have to follow her act, and it is a tough act to follow, but I will do my best. Welcome to all of those who are listening on the streaming presentation. We're going to discuss the origin of life, another step forward. What is life? Life has been defined in many ways, and we're going to look at life has been defined in many ways, and this particular list represents major general properties of life as defined by the biological life sciences. It was actually selected from a 10-page document on Wikipedia defining life. We will concentrate primarily on the fourth item, molecular biology and biochemistry, since it represents an area of most active research. New information is emerging primarily as a result of the rapid development of new techniques and tools, enabling us to observe life at a molecular level. The rapid expansion of our knowledge has not simplified the search for the origin of life, for we now recognize that living cells and life processes are far more complicated than we previously thought. No longer can we view a cell as a sack of cytoplasm, as was commonly thought in Darwin's day. Every living cell contains millions of different proteins, is highly compartmentalized, contains millions of nanomachines that operate at unbelievable speeds and accuracies, enabling them to synchronize thousands of biochemical reactions occurring simultaneously. Of the many questions that might be considered pertaining to the origin of life, we will concentrate on the following three. First, what scientific avenues are available in our search for the origin of life? An event that apparently occurred once, giving rise to a single evolutionary family tree based on a genetic code capable of giving rise to all of the interrelated forms of life known. Secondly, what scientific evidence is there to support a gradual, progressive march toward increasing complexity of cells, leading to the synthesis of, on average, 10,000 protein molecules, averaging 400 amino acid molecules in a precise sequence, in which there are, on average, 1 million exact copies of each protein molecule, all continuously functioning in the average cell. Thirdly, given the presence of irreducible complex structures in virtually every cell, how is it possible for random mutations causing small incremental chemical changes to generate a double helical biochemical molecule totaling more than 3 billion base pairs in highly encoded a biological encyclopedia instruction manual for building and operating all of the living organisms that we know. Particularly the one that each of us occupy, which consists of about 37.2 trillion cells in the average human that work purposefully with conscious awareness and an onboard recording system that can record and recall all the important events of a lifetime. We will now consider the scientific pathways available in the search for the origin of life. This line of investigation is an attempt to show that simple chemistry may have produced a series of organic compounds that in turn resulted in the formation of prebiotic compounds that eventually led to a simple biology that over a long period of time resulted in the simplest forms of life. Two major scientific pathways have been pursued to date. The first was put forth in 1923 independently and simultaneously by Alexander Operum, a Russian biochemist, and J.D.F. Haldane, a British scientist. Their concept became known as abiogenesis, in which they proposed that conditions present on early Earth could have been conducive to spontaneous formation of organic compounds based on simple chemistry. They postulated that water vapor, hydrogen, and methane were present on early Earth in what was generally referred to as primordial soup. Operum was convinced that coastervates, suspended colloidal particles in the primordial soup, played a prominent role in the abiogenesis process. Subsequently, however, it was documented that they are inorganic colloidal particles and are unrelated to organic chemistry. It should also be recognized that abiogenesis is distinct from spontaneous generation, thought to have been the origin of life from the 17th into the 19th centuries. The Miller-Urey experiment was the first serious scientific attempt to verify the Operum-Haldane theory of abiogenesis that occurred some 3 decades later by two American chemists, Stanley Miller and Harold C. Urey, in a basement here in the University of Chicago, a basement laboratory, in which they performed the now famous Miller-Urey experiments starting in 1953. Their experiment was simple, consisting of a series of flasks connected by a tube forming a circuit. One flask contained warm water, ammonium, methane, and molecular hydrogen, all of which were thought to have been present in the early Earth environment. The water in the flask was heated gently to mimic the warm, shallow inland bays and lagoons. A second flask, rigged with tungsten electrodes, produced electrical sparks thought to simulate lightning strikes, was connected with glass tubing to complete the circuit. The experiment was run continuously for a full week, at which time the solution became brown and the flask became coated with a black, tarry, oily-like substance. Analysis of the fluid content yielded several amino acids, including glycine and alanine. The announcement of these findings generated a great deal of enthusiasm and captivated the imagination of many origin of life scientists, resulting in a large series of similar experiments with slight variations thought to more accurately simulate early Earth conditions. These did result in the synthesis of a relatively large number of organic compounds. However, a far larger number of organic compounds essential for life remain missing, and this line of investigation has largely been abandoned. Their approach has become known as the metabolic pathway. The second scientific pathway toward the origin of life is known as the ribonucleic acid world theory. The RNA world theory proposes that early life originated during a time when RNA was the predominant prebiotic biochemical molecule, which then gave rise to early life. Its history goes back to the 1960s when Leslie Orgel, Carl Woese, and Frank Crick hypothesized, based on the folding characteristics of RNA, that it may have been a major early life molecule, perhaps a sole macromolecular basis for the evolution of the machinery of life. It was the work of Sidney Altman and Thomas Keck, with their discovery of the catalytic properties of RNA, for which they were awarded the Nobel Prize in Chemistry in 1989, that provided a biochemical basis for seriously considering RNA as a primary early life molecule. The catalytic properties enable self-replication when combined with the potential of an information molecule, greatly enhance the likelihood of its association with early life. When one When one considers that virtually all forms of life depend on DNA to encode their genetic information, and that the encoding system is universally functional throughout both the plant and animal kingdoms, one cannot escape the conclusion that the origin of life is linked to the source of information that provides the basic directives for all living organisms. Based on phylogenetic considerations alone, searching for the origin of life among the information molecules, which are RNA and DNA, is clearly a step forward from the original search along the metabolic pathway pursued by Miller and Urey. One of the leading proponents of the RNA world theory is Jack Szostak, professor of genetics at Harvard Medical School. He has lectured extensively on this topic. He has recognized that a number of the important protein molecules have associated nucleotides, which he proposes represent relics of the RNA world. Examples are acetyl-CoA, vitamin B12, and the extensive RNA molecules that make up ribosomes. These are interesting theoretical considerations, but do not address the problems of stereospecificity, the combinational problem, nor does it provide any explanation for the origin of the universally encoded information in the DNA comprising every genome. Perhaps the simplest way to understand the concept of stereoisomerism, or chirality, is to look at one's hands and note that although each has 4 fingers with an opposing thumb, the shapes of the hands, being mirror images, fit into small spaces very differently. For example, the right hand does not fit into the left glove, nor vice versa. Like left and right-handedness, chiral biochemical compounds occur in 2 forms, mirror images of one another, but otherwise consist of the same exact sequence of atoms and chemical bonding characteristics. Their chemical formulae are identical, but their spatial configuration differs. Since so many biochemical reactions involve compounds fitting together like a lock and key, different external configurations will result in different biochemical activities. Chiral compounds are frequently found in biochemistry because many compounds contain carbon atoms that have 4 possible bonds. If each bond is connected to a different moiety, such as illustrated in this slide, then a chiral center exists, as illustrated on this slide. Under ordinary chemical circumstances, 2 stereoisomers, each with its separate spatial configuration, will occur, producing a mixture of stereoisomers known as a racemic mixture. And this occurs in all cases where just simple chemistry is depended on. For reasons not understood, stereoisomers in living organisms seem to follow strict conventions such that all amino acids are left-handed and all sugars are right-handed stereoisomers. Since almost all proteins synthesized by living organisms is done by enzymatic action, in which nanomachines precisely build proteins atom by atom and molecule by molecule, and can therefore produce stereospecific compounds consistently. When biochemical compounds are produced by non-living chemical processes, stereoisomerism becomes a major consideration, such as in the pharmaceutical industry. The FDA's policy is to treat each stereoisomer as a separate drug. A few examples establish this point. Naproxen, or Aleve, a commonly used NSAID, is a chiral drug in which the right-handed stereoisomer has excellent NSAID activity, whereas its left-handed isomer is highly toxic to the liver. Ketamine is an anesthetic, whereas its isomer is a hallucinogenic drug. You can imagine what might happen in the OR if those 2 got mixed. It wouldn't be a pleasant scene. Darvon relieves pain, whereas its stereoisomer is a cough suppressant. And there is no known solution to the racemic mixture problem. Now we will address the dilemma of the origin of encoded information in DNA. This is a picture of James Watson and Francis Crick being congratulated by Macklin McCarty for their work in accurately delineating the molecular configuration of DNA. The double helical configuration of the DNA molecule is modeled on the right. and you can appreciate that that's a somewhat dated model. With the discovery that 2 DNA strands were on the inside, based on X-ray crystallography images produced by Rosalind E. Franklin, Watson and Crick were enabled to correctly identify the double helix configuration of the DNA molecule in February of 1953. On February 28, Crick interrupted the patrons at lunch in the Eagle Pub in Cambridge, England, to announce that he had, quote, discovered the secret of life, end quote. In 1962, Watson and Crick were awarded the Nobel Prize in Chemistry, shared with Maurice Wilkins, who was a research colleague of Rosalind Franklin, whose photograph of the X-ray crystallography image of DNA played a pivotal role in enabling Watson and Crick to correctly identify the double helix configuration. Rosalind Franklin died of ovarian cancer prior to the awarding of the Nobel Prize, and it's reported that she never knew the important contribution she made to correctly identifying this most important molecular structure in all of biological science. Crick went on to recognize the central dogma of molecular biology. RNA and DNA have many similarities. Both are primary genetic information carrying molecules in, in all of biology. Excuse me. Both are long polymers forming a helical configuration. DNA normally displays a double helix, whereas RNA is usually single. Both are similar in structure, consisting of a phosphate group linked to a phosphodiester bond to a pentose sugar that in turn is linked to a nitrogen and carbon containing ring structure commonly referred to as a base. The pentose in RNA is ribose, and the pentose in DNA is desoxyribose. There are 4 bases in each, in which in DNA are cytosine, guanine, adenine, and thymine. And in RNA, they are similar except uracil replaces thymine. Adenine and guanine are purines, whereas cytosine and thymine and uracil are pyrimidines. A most remarkable feature of DNA is that the base pairs are located on one of the double helical spirals, is accompanied to those in the other, such that cytosine is always located opposite guanine, forming a hydrogen bond, which Marjorie pointed out to you as being very important bonds in biology and in living organisms. And in this case, these hydrogen bonds are what hold the 2 strands together and what can unzip and rezip so readily. Let's see, just a second. The structural arrangement contributes greatly to the remarkable stability of the DNA molecule, in which it can remain stable for many thousands of years. One other important consideration is that the complementary organization of the base pairs on the DNA molecule are, from a chemical point of view, identical. The chemical bonds and hydrogen bonds linking the base pairs to the double helix, as well as to each other, are all exactly equal for each base pair. Therefore, there is no chemical preference for the location or sequence of the base pairs on the DNA polymer. This raises a profound question. If chemical bonding is not responsible for the sequential arrangement of the base pairs along the double helical spine that encodes the information directing the synthesis of all of life's essential proteins, how was the encoded information derived? Where did it come from, and how did it get there? The universal genetic code is based on the sequence of base pairs on the DNA strand. There are only 4 letters in this unique genetic alphabet, corresponding to the 4 nucleotides, C for cytosine, G for guanine, A for adenine, and U for uracil, that make up the base pairs of the RNA molecule. These 4 letters make up the alphabet on which the genetic code is based. The code uses only 3-letter words based on the sequences of the nucleotides. With a total of 4 letters, the number of possible combinations of 3-letter words is 64. That's 4 to the 3rd power. I think you'll agree with that. Each set of 3 nucleotide sequences is known as a codon. There are 61 codons to encode the 20 required amino acids. The remaining 3 are stop codes indicating the end of the strand of amino acids of the protein under construction. The number of codes available for each of the 20 amino acids varies between 1 and 6. Methionine, indicated in this illustration by the black triangle, is uniformly located at the beginning of the protein polypeptide chain. This is true of all prokaryotes and eukaryote cells. The end of the polypeptide chain is signaled by a stop code identified by a black circle. The sequence of codons composing the entire polypeptide chain is referred to as a reading frame. Thus, each reading frame begins with the code for methionine, which is AUG, and ends with the stop code, one of the following 3, UGA, UAA, or UAG. The most remarkable feature is the fact that a given codon encodes the same amino acid in essentially all living organisms, including bacteria, yeast, fungi, animals, and plants. As a result, a universal genetic code has emerged based on genetic studies across a broad selection of living organisms. The universal genetic code is defined as, open quote, the instruction manual that all cells use to read the DNA sequence of the gene and build a corresponding protein, end quote. There are a few scattered exceptions to this rule that mainly occur in mitochondrial DNA that is thought to have resulted from later evolutionary developments. There is no evidence to support a major overhaul or alteration of the general genetic code since the earliest function. And again, another quote from a major textbook. The discovery that virtually all forms of life use DNA to encode their genetic information and also use nearly identical genetic code implies that all forms of life descended from a common ancestor based on the storage of information encoded in nucleic acid sequence. Of the many universal genetic code charts available, This pie-shaped chart is among the most elegant and simplest to use. The first letter of the codon is in the innermost circle of the chart. The second letter is in the second ring, and the third letter is in the third ring, forming 3 concentric circles. The abbreviations for the encoded amino acids are located around the rim of the chart. Again, because of the universal nature of the code, the same codon encodes the same amino acid, whether it is in an early aquatic plant of 400 million years ago, a reptilian of 100 million years ago, a mammalian of 50 million years ago, or a human being of 500,000 years ago. Replication of DNA is the first step in the central dogma of molecular biology. Before DNA replication can occur, specific sites known as replication origins must be identified on the DNA molecule. Specific helicases, the blue wedge identifies it, on the DNA molecule. Specific helicases bind to the site and the beginning of the unwinding process of the parental DNA double helix before replication can begin. Since the 2 strands of the double helix molecule are anti-parallel, the DNA polymerase can grow the new strand only in the 5' C' direction, duplicating the leading strand without interruption. Replication of the lagging strand, however, is much more complicated and consists of forming short segments in the opposite direction, in which an RNA polymerase called primase starts the replication process by forming a short strand of RNA primer, which becomes attached to the newly formed replicated DNA strand, the combination forming an Okazaki fragment. Okazaki fragments that are subsequently joined together by DNA ligase, reestablishing continuity of the newly synthesized lagging strand, again in the 5' 3' direction. The above described diagram indicates unidirectional replication of DNA. However, there are multiple studies supporting simultaneous bidirectional DNA replication, but that would be very hard to show on a diagram. The large green colored ring surrounding the double helix on the right side of the slide, labeled topoisomerase, provides an important function in controlling or eliminating left-handed supercoiling that may result from the double helix when the double helix is unraveled during DNA replication or RNA transcription. Since the ends of the double helix are not free to twist, uncoiling of a relatively long stretch of DNA causes left-handed supercoiling. The topoisomerase may at times relieve the supercoiling by cutting one of the strands and then rejoining them after proper coiling has been reestablished. It should be noted that the helicase is a large, complex macromolecule, and not shown are a series of other slightly different DNA polymerases that are also required. So this business of replicating DNA that occurs every time a cell divides is rather complicated. The concept of the central dogma of molecular biology was first proposed by Francis Crick, who recognized the universal continuum. It occurs in 3 stages: The first stage depicted in the previous slide is replication of DNA. The second stage is transcription, where messenger RNA is copied from the DNA. And the third stage is translation of the messenger RNA to synthesize protein in the ribosome. This slide graphically depicts the central dogma of molecular biology in slightly greater detail. The top line illustrates a segment of DNA molecule that is undergoing transcription of the gene to form a molecule of messenger RNA. The beginning of the transcription process occurs as the promoter region on the DNA strand and continues to the right. Initially, the strand is copied in continuity. Subsequently, the introns, depicted in black, are cut out, and the remaining exon portions in the molecule, depicted as red, are spliced together to form the messenger RNA molecule. This process is labeled messenger RNA processing. The next step consists of translation of the messenger RNA into a protein molecule that occurs in the ribosome. The long protein polymer is then folded to form an active protein molecule. And this is formed by the chaperones that Marjorie mentioned. In summary, the central dogma of molecular biology involves replication of DNA, followed by transcription of messenger RNA that is translated into protein. Note that the protein molecule begins with the amino acid methionine, which always occurs according to the universal genetic code. It is also, okay, we will now observe a brief YouTube display of how this actually occurs in real time.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Pretty impressive cells we have working for us, all 32 trillion of them. Now we're going to look a bit at cellular histology, and we'll review a few of the highly complex biological, physiological processes that are fundamental to all living organisms as we consider the implications of our third question. Living organisms are generally grouped into 2 cell type classifications, prokaryotes and eukaryotes. Prokaryotes do not have defined nuclei and are much smaller than eukaryotes, approximately 1,000th their size. Eukaryotes compose the vast majority of the cells of living organisms, including yeast and all multicellular organisms, whereas prokaryotes primarily compose single-celled organisms such as bacteria and archaea. Other major differences are the presence of organelles in eukaryotes, such as mitochondria, Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, cytoskeleton, and centrioles, which are completely absent in prokaryotes. Each organelle has specific functions and occupies specific compartments within the cell, such that related functions are grouped together, much like a manufacturing facility might be organized for efficiency. No known intermediate forms or evidence of an evolutionary bridge between prokaryotes and eukaryotes exists. The sodium-potassium pump is one of the most important basic nanomachines in all living organisms. It is a large, complex, macromolecular protein extending across the cell membrane, capable of active transport between the intra and extracellular compartments. The cell membrane is a bilayered structure, as depicted in the slide. It can pump 3 sodium ions out and 2 potassium ions into the cell with each cycle, requiring 1 molecule of ATP. Thus, the intracellular fluid becomes 1 unit charge more negative relative to the extracellular fluid. In addition, the positive concentration gradient of the potassium ions across the cell membrane is increased by 2, and the negative concentration gradient of sodium increases by 3. Other significant functions of the sodium-potassium pump include maintenance and control of the cell volume by adjusting osmolality, and importation of glucose and amino acids and other nutrients into the cell by use of the protein gradient. The average resting transmembrane electron potential measures minus 30 millivolts in most somatic cells. However, in neurons, it measures in the range of 70 to 80, that is minus 70 to 80 millivolts, since electron potentials are required for neurotransmission. The transmembrane electrical potential is an essential feature of all living cells. The amount of energy required by the sodium-potassium pump to maintain the resting cell electron potential is calculated to be in the range of 30% of the total cell energy requirements. The sodium-potassium pump was first reported in 1957 by Jen Christian Sue, and 40 years later, in 1997, he was awarded the Nobel Prize in Chemistry. And I'd also like to mention that it's been estimated that in the average person, one requires 60 kilograms of ATP per day. That's more than one's total body weight. So that explains why some of these nanomachines need to work at the speed they do. And we will see that in just a second. Since the sodium-potassium transmembrane pump requires energy, a second large macromolecular nanomachine capable of generating ATP is required to maintain the transmembrane potential. ATP synthase is the commonest source of ATP throughout the animal kingdom. They are built by ribosomes, the basic protein factories in living cells. This brings us to the fundamental question related to the concept of irreducible complexity that Charles Darwin proposed. In The Origin of Species in 1859, he wrote, open quote, "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, but I cannot find out any such case." End quote. Here we are faced with the dilemma that Darwin anticipated. That is the extremely remote possibility of large, irreducibly complex biological structures coming into existence by a series of gradual advances based on random mutations. He had no way of knowing that every cell would require irreducibly complex nanomachines to maintain even the very most basic life support system, such as a resting cell transmembrane electrical potential. As illustrated in the slide, it is the large macromolecular nanomachine consisting of multiple protein molecules forming large complexes with multiple moving parts. There is a rotor, a stator, a drive shaft, and a series of cams, in many ways similar to an electric motor in that it spins like an armature. However, the rate at which it spins is 134 revolutions per second, or 8,040 revolutions per minute, faster than most of your electric motors. Details of its operation, such as its conformational changes in specific amino acids, electron flow pathways that drive the motor, and the arrangement of the various parts are included in my accompanying paper. And now we will look at a brief video.
It's been called one of the wonders of the molecular world, an amazing nanoscale machine. ATP synthase is a high-tech micromolecular power generator inside the cells of your body. It generates adenosine triphosphate, or ATP, an energy molecule that provides fuel that every one of your cells needs to function. Without this fuel, your cells will cease operation, and so will you. ATP synthase works like a rotary engine. The barrel-shaped rotator is composed of 10 to 15 protein parts called subunits. The rotator spins around, transmitting mechanical energy into the drive shaft of the machine, which helps make ATP. This drive shaft has a specially placed bump that opens and closes parts as the drive shaft spins around. This bump opens special protein subunits on the bottom of the machine. When the bottom subunits open, a spent energy molecule called adenosine diphosphate, or ADP, enters the machine. The mechanical motion causes the ADP to bind with an additional phosphate group, creating the ATP energy molecule. And the ATP drifts off into the cell, ready to power some biomechanical reaction. The ATP synthase machine has many parts we recognize from human-designed technology: a rotor, a stator, a drive shaft, and other basic components of a rotary engine. The ATP synthase is one of thousands of elegantly designed molecular machines inside your cells that make your life and all known life possible. ATP synthase, an example of intelligent design.
And this sort of thing is going on in every one of our 32 plus trillion cells, constantly, 7/24, no relaxation, no stopping. Now I'd like to just briefly mention a couple of the major signaling systems that enable our cells to send signals from one to another, to work together, and so forth. One of those major ones is the G protein coupled receptor, which is known as GPCR, sort of a tongue twister. And the point is that no cells can actually survive in isolation. Intracellular communication is a basic property of all living cells. Humans have more than 1,000 unique GPCRs in each cell, and each is very specific for a precise cell function. GPCRs are positioned in the cell membrane, providing sensory input from the external environment. These represent the targets for some 30 to 50% of all medications. In 2012, the Nobel Prize in Chemistry was awarded jointly to Brian Kobilka and Robert Lefkowitz for their GPCR research. The most characteristic structural feature of the GPCR is that they have 7 transmembrane receptors, which are illustrated here by the azure blue objects, which are actually transmembrane, so they can transmit a signal from the external to the internal portion of the cell. There's 6 different stages here in this process, which I will not go through, but I would like to just mention Well, I'd like to mention that the so-called fight or flight response that can occur when one is startled or in danger is signaled by this process, and it causes like changes in your heart rate. It causes release of glucose from the liver. It causes dilatation of the arteries to the muscles, which enable you to respond actively to a potential danger. So this system is responsible for all of those cellular changes that occur at a moment's notice. But the one probably most fascinating aspect of it is that this process, a modification of it, is what enables us to see. It's this signaling system that is located in the retina of the eye. The rhodopsin in the rods and cones act as the GPCR molecule, and the transduction signal is a photon instead of some other messaging device. And of course, the final message is a neuronal impulse carried to the optic cortex, where visual images are interpreted. And in this case, the signal transduction system, it is essential that the system return instantaneously to baseline so that it is ready to receive a fresh signal indicating motion of the object being observed. So as a result, the normal base level is restored within 50 milliseconds. This is another system which is interesting because it was the first non-gene signaling system that affects the expression of genes. It occurs in E. coli, and happens because E. coli has a preference for glucose for its energy source. However, if lactose is available, it will utilize lactose. But lactose will actually block the system if glucose is present. So it's a very interesting event there. And then there are many hormonal controls. They arrive extracellularly. They link with a hormone receptor complex, which allows them to actually enter the nucleus where they have access to genes and can therefore direct the production of a protein such as growth hormone or thyroxine or one of those many hormones that are so important for us. In conclusion, new discoveries are constantly being made, requiring revisions to previous conclusions or theories. Amino acids are all left-handed, and sugars are all right-handed in living organisms, where chiral centers exist. Simple chemistry generates racemic mixtures only. Complexity of biochemical processes and a number of macromolecules that work perfectly together, as in many nanomachines, are irreducibly complex, a situation incompatible with neo-Darwinian theory. Gradual random mutations have not been shown to be able to produce the complexity of observed living functions. For example, no intermediate life forms have been found that bridge the gap between prokaryotes and eukaryotes, yet they use the same DNA transcription mechanisms. As an average cell, tens of thousands proteins comprised of hundreds of amino acids need to be assembled in a precise sequence and often work in one precise way with a different protein to be of any evolutionary benefit. Random formation of these would result in millions of partial dysfunctional protein cells, which we do not see. The Universal Genetic Code is the instruction manual that all cells use to read DNA sequence of the gene and build corresponding proteins. There is no evidence of a major revision or alteration of the code since its earliest function. The encoded information exists at the onset or prior to evolution. The source of the information remains a major dilemma. Thank you very much. And I'd like to, first of all, I'd like to thank my lovely wife who put up with me for the many hours I spent preparing this. And I'd also like to thank my son, who helped me a great deal with the technical aspects and helped to put this slide deck together. And I thank each of you for the inspiration that you've given me. I really enjoyed reading your papers, and I think that we're on a wonderful track here in seeking to communicate with our scientist friends out there these many fascinating things that are happening around us. Thank you very much.